now and next speaker, and by the end we will have a Q&A session. So it's my pleasure to invite our next speaker, Professor Dinara Galiyeva, professor from Medical School Nazarbayev University, and Dinara will talk about survival analysis as a key tool in quantitative medical research. Please, Dinara, floor is yours.
Thank you very much, Professor Konesh. Just let me share my screen.
Do you have access for the publication? Okay.
I have, yes. Okay, so let's start. So why I decided to talk about survival analysis today? Because survival analysis is one of the key tools in many research. So basically, all clinical fields are interested in assessing survival of patients, survival of different groups of people, of patients taking different drugs. And I think many clinicians have the research question in mind to assess whether one treatment is better than another in terms of survival probabilities. So we will talk about that. So there are different research questions. So for example, the prognosis of graft rejection, patients taking different drugs, what is survival probability? How does comorbidity affect the access to transplantation? So all those sort of questions require the analysis of survival probability. So to answer these questions, in terms of prognosis estimation, treatment comparison, and time to event outcomes, we use Kaplan-Meier, and we will talk about that today. So I will explain what is Kaplan-Meier method, how we compare different groups between each other, how we visualize or how we plot survival probabilities, and how we can test the hypothesis. So one of the primary aim of the survival analysis is the modeling and analysis of time to event data. So basically, the data that have as an endpoint the time when the event occurs. And event can be not just death. Of course, mortality is one of the commonly used endpoint in many research studies, but you can also study other events, not necessarily death. So you can study, for example, myocardial infarction as an event, recovery of renal function, first renal transplant, for example, graft failure, or time to discharge from the hospital. So all of these events can be the endpoint in the survival analysis. But we need to understand that not during the observation period, not all patients will experience the event. So some patients will not experience the event during the observation period, and this is called as censoring. So this is one of the important points to understand. So during the observation period, the censoring happens when patient didn't experience the event, and he could not experience the event. So we need to understand that during this follow-up period, we don't know if the patient developed the event or not. We only know that during this time period, the event didn't happen. And this is called the censoring. And censoring can also occur when the follow-up is finished and when we lose the patient. So loss to follow-up happens in all clinical research, in all clinical studies. And in this scenario, when we lose the patient for follow-up, we also censor the patient. The other point where we censor the patient is when, for example, competing event happens. What is competing event? So competing event is another event that happened before the event of interest. So it calls the competing event. For example, if you follow the patient from the start of renal replacement therapy and you are interested in death from myocardial infarction, but your patient died from, let's say, malignancy. So this is the malignancy is competing event, and you need to censor the patient when he experienced the competing event. So it means that during the follow-up, the event could happen and the event could not happen during the observation period. When event doesn't happen, we treat it as censoring. And censoring happens when we lose the patient for follow-up or the patient experience the competing event or the study ends. For example, you set the end of the study, let's say 31st December 2026. And this is the end of follow-up where you need to censor the observation. So one of the examples. So let's say you have the cohort of patients on renal replacement therapy. You recruited them from 1996 to 2000, and you observed them. You followed them till December 31st, 2005. So this is the end of follow-up period. And your event of interest is death while on renal replacement therapy. So the censoring takes place at recovery of renal function, loss to follow-up, or December 31st, 2005. So if we visualize it, this is an example. So let's say that out of these 8 patients who were observed, who started RT, event of interest, so the death on renal replacement therapy happened among 1, 2, 3, 4, 5 patients. 3 patients were censored. So this is the visualizing how it happens. So the event of interest. And Kaplan-Meier method is one of the tools that we use to estimate survival probabilities, and we handle censoring data. So let's say another example. So let's say 50 patients started renal replacement therapy. Survival time started running from the moment of the start of RT, and patients were followed until death. So this is the all-cause mortality was the event of the interest or censoring, and we use Kaplan-Meier method to estimate survival probability. And this is the table that usually the statistical software that does this. But I would like to explain the calculation. So here are all the patients who were recruited and were at risk of death. The first column is the time in days. So let's say the first patient died on day 34 of the observation period, and the proportion surviving on this day would be 49 patients divided by 50. It's 0.98, and cumulative survival probability is 0.98 multiplied by the cumulative survival of previous period. So in this case, the previous period was 1 because no one died on day 0. So the cumulative survival would be 0.98. And then when each death happens, you need to calculate the survival probability. The cumulative survivals, sorry. When censoring happens, so for example, on day 57, one patient was lost to follow-up, so he was censored. It means that out of 47 patients, 47 were alive, so it will be 1. And survival probability would be the same, so no deaths were observed. And the same happens with all the recorded deaths and all the recorded censoring. So this is the Kaplan-Meier survival table when we calculate death and censoring patients and calculate cumulative survival. So once we calculate cumulative survival, we can plot it. Using you're using these Kaplan-Meier survival plots, and you see that every point, so the survival graph goes down, and every death, with every death, the graph goes down. With the censoring, the graph stays at the same level. So, and it shows the visualizing, it visualizes the survival probability. So the same information from the table translates to the graph. So in the stepwise survival calculation, we calculate survival probabilities and cumulative survival. So how we report? Usually we report survival probabilities at year 1, for example, at year 2, year 5. And for example, in this example, survival probability at year 1 was 79%, at year 2 was 70%, at year 5 was 40%. So based on the table here, so we can take the numbers from this table. We can also compare survival probabilities between different groups. To be able to do that, we can use the Kaplan-Meier table at the specific time point and to see whether at this time point it's different or from between two groups. But this is not very convenient because we're not taking into account the whole follow-up period. For that, we use the log-rank test. So once we split or stratified by different groups, we can do statistical testing. So we can test the hypothesis. So basically, the hypothesis is there is no difference. There is no statistically significant difference in survival probabilities between two groups. And we test this hypothesis using the log-rank test. And log-rank test takes into account the whole follow-up period, not just the specific time point. So, for example, here, when we compare different survival between different modalities, so for example, patients started on renal replacement therapy using hemodialysis or peritoneal dialysis, we can compare survival between these two groups. And we use log-rank test to be able to assess the significance level. So in this case, p-value is 0.04. So it means that this is the statistically significant. There is a statistically significant difference between two dialysis modalities. And we also can present differently. So we can present either as survival plots or mortality plots. So basically, this is the same information, but presented either as survival or either as recorded mortality. The information is basically the same. It just depends on the authors or depends on you. How would you like to present the numbers? So there are limitations of Kaplan-Meier methods and log-rank test because, of course, it is very popular. Kaplan-Meier methods is one of the popular tools to assess survival probabilities. And with log-rank test, it can test the hypothesis and to test their significance. However, if you would like to adjust for different confounding factors, for example, you would like to adjust for age or adjust for sex, ethnicity, some of the regional differences, for example, you cannot do that with a log-rank test because it takes into account only one variable. You can stratify that. So, for example, do the whole analysis on male sex or female sex, for example, but not able to adjust. So for that, you will need to do the Cox regression model. And Cox regression model, it's multivariable model, which can apply the adjustment for different confounding factors. So also you cannot see the confidence intervals with the log-rank test, so you cannot estimate the significance level. So again, for that, you will need to use Cox regression model. So in the Cox regression model, of course, it's more, it gives us more information. It gives the opportunity to adjust for different confounding factors. It gives us the estimation so that we will see the hazard ratio and we will be able to assess the power, having the 95% confidence intervals, for example. To conclude, Kaplan-Meier is one of the powerful tools to describe and to assess survival probabilities in patients. It estimates survival. Log-rank tests test the hypothesis that there is no significant difference between two groups. And log-rank test helps to estimate, to test this hypothesis, not to estimate, to test the hypothesis. And if you need further, more deep analysis, multivariate analysis, or adjustment for different confounding factors, you will need to use Cox regression analysis. Okay, that's it for the survival from me.
Thank you, Professor Dinara. Thank you very much. Okay, the next